Hello everyone. In this video, we are going to discuss different types of amplifiers. So, what in the previous video we have discussed what is the need for bioamplifiers. So, the biological signals which has been picked up from the patient's body are weak in amplitude as well as in the frequency. In order to boost up the biological signals, we go with bioamplifiers. So, as we have seen, there are five different types of bioamplifiers. They are differential amplifiers, operational amplifiers, instrumentation amplifier, chopper amplifier, and isolation amplifier. Let's see each and every bioamplifiers in detail. So the first one is differential amplifier. So, differential amplifier, as the name indicates, here we have an operational amplifier. The differential amplifier, as the name indicates, the output will be the difference of two inputs. So, V out is your output, and V1 and V2 are the two inputs. V out will be the difference between the two amplifiers, and it would be multiplied by a gain where AD is called as to be the differential gain of an amplifier, and if V1 and V2 are equal to 0, the output voltage will be equal to 0. So, differential amplifier, as the name indicates, the output voltage will be the difference of two input signals. If two input signals are equal, the output voltage will be 0. So, the final output voltage is directly proportional to the difference between the two input voltages. And the second type of bioamplifier is operational amplifier. And this is the operational amplifier symbol. Here we are going to use the 741 IC 741, where we have a non-inverting terminal and an inverting terminal and a positive power supply and a negative power supply and then final output. So the ideal characteristics of an op amp, the operational amplifier can also be called as to be the op amp. And so the ideal characteristics of an op amp is the final output voltage should be of infinite, input resistance should be infinite, whereas the output resistance should be zero. As usual, the major thing is it should have a free from noise and drift, which should be of zero noise, and pass supply rejection ratio should be zero, and CMR should be very, very high in order to maintain the bioamplifier. Very high in the sense it is going to be an infinite. So the operational amplifier can be of two types. One is inverting amplifier and the other one is the non-inverting amplifier. So the first one is inverting amplifier. So as we have seen, so the inverting amplifier, the input is given to the inverting terminal. So the output will be 180-degree out of phase with each other. So whatever the input is given, the output will be 180-degree out of phase with each other. If the input is going to be the positive, the output will be of negative. If the input is negative, the output will be of positive. It will be in the reverse order. So totally it is 180-degree out of phase with each other. The second type of amplifier is non-inverting terminal, where the input is given to that of the non-inverting terminal, which is a positive terminal. So whatever the input is given, the output will be the same. So the both the output and the input will be in phase with each other, will be in phase with the input. So if the input is positive, the output will also be positive. If the input is negative, the output will also be negative. And the gain of the non-inverting amplifier is 1 plus R2 by R1. R2 is nothing but the feedback resistance and R1 is going to be the input resistance. And the third type of biological amplifier is instrumentation amplifier. So far we have seen what is differential amplifier, operational amplifier, and the third type is going to be the instrumentation amplifier. So this is an instrumentation amplifier where we will have three op amps connected with each other. The two op amps, the input are given to a non-inverting terminal. So whatever the input is given, the output of the amplifier A1, the output of the amplifier A2 will be in phase with that of the corresponding input. So if it is output of A1 will be in phase with that of V1, output of A2 will be in phase with that of V2. And finally, these outputs are given to a third operational amplifier, and this operational amplifier will be behaving as an differential amplifier. So, the output of a differential amplifier, as we have studied in the previous slides, so the output of the differential amplifier will be equal to the difference between the two inputs. And very important point to be noted in instrumentation amplifier is it should have a high CMRR, common mode rejection ratio will be of very high, which is an infinite CMRR. So totally here we have three op amps, two for the inputs and two the inputs will be given to the non-inverting terminal, and the output of that is given to the third op amp, which is in differential amplifier. And the fourth type of amplifier is going to be the chopper amplifier. So, chopper amplifier, the major requirement of a bio amplifiers, as we have discussed in the previous video, it should be free from drift and noises. As I have told you, drift is nothing but the thermal disturbances which has been taking place inside the room as well as the outside the room. So it could be of natural disturbances or any power fluctuations or anything which taking place inside the room is called as to be the drift. So this chopper amplifier acts very good in reducing the drift as well as noise in case of the bioamplifiers. The chop the word it is used to sample the data. So let's now just go in detail how the basic chopper amplifier works as a bioamplifier. So this is a basic block diagram of a chopper amplifier where we'll have a chopper. Whenever a DC input signal is given to a chopper, so this chopper it is used to sample the data. So it samples the data and we'll get the output of the chopper as to be a pulsed DC. Here we could see which is going to be the pulsed DC. And internally we'll have an digital to analog converter where this DC signal is converted to an analog signal. The analog signal are very less in amplitude or low in amplitude. So in order to boost up the signal, here the AC amplifier has been used. Analog amplifier will be used and the final output of the analog amplifier will be of amplified analog signals. So till this it is going to be the modulation part. Whatever the input is given, it is going to the DC input is going to get converted to an AC where chopper it is used to sample the data and AC amplifier it is used to amplify the low analog signal and finally we'll get back the amplified AC signal. So this modulated output is given as an input to a demodulator. So, as we are all aware, the function of a rectifier, the rectifier, it is used to convert AC to a DC signal. So, this demodulator here, the rectifier is used wherein takes the amplified AC signal, it converts back to the original DC signal, which is nothing but the input to an modulator. So, this is a basic block diagram of a chopper amplifier. And so far we have discussed about differential amplifier, operational amplifier, instrumentation amplifier, chopper amplifier, and now we will move on with the last type of the amplifier, which is going to be the isolation amplifiers. So, the isolation amplifiers are also known as pre-amplifier circuits, isolation circuits. So the major requirement, as we have discussed the requirements of a bioamplifiers, each and every requirement has been satisfied by certain amplifiers. In order to avoid the drift, we go with chopper amplifier. In order to avoid the impedance matching of the circuit, we'll go with instrumentation amplifier. In order to avoid the electric shock or accidental shock, we'll go with an isolation amplifiers. So approximately 1012 ohm of insulation is provided between the patient and the power line which is placed inside the hospital. So this isolation amplifier is helps to prevent the electric shocks between the patient and the instrument. So the basic symbol of an isolation amplifier, it is going to be we have an input connected to the positive terminal, an output to the negative terminal, there is an isolation between the amplifiers. So this is an isolation amplifier which is internally isolated. So next we'll just move on with the basic block diagram of an isolation amplifier. So this is going to be the modulated part. This is the demodulated part. Whenever the input is given to an amplifier, it is going to get the amplified and it is passed through the modulator. And this modulator is now passed through that of an LED or a transformer or a primary transformer. So this LED with sensor, this LED could be a photodiode or a photodetector which senses the light and it holds there for a certain duration of time. That is, your isolation barrier will be there, and after certain duration of time it is transformed through a photodiode or a photodetector or the secondary of the transformer, and then finally it is transmitted to the demodulator where the rectifier will be used as a demodulator. And if the amplitude are very low in amplitude, it gets amplified and we will get back the original input signal. Whatever the original input signal has been given to a modulator, we will get back the original input signal as the output of the demodulated circuit. So this is an isolation amplifier. So each and every amplifier is capable of performing a certain requirements of a biological amplifier. The isolation amplifier is used to prevent the electric shock. Chopper amplifier is used to prevent the drift and noises in the circuit. An instrumentation amplifier could be used as an impedance matching circuit. An operational amplifier, it is used to do the operational of mathematical operations. Differential amplifier, where the output would be directly proportional to that of the input, difference between the input and the output voltages. Thank you.